Welcome back to Biotech Wonders, the captivating channel dedicated to biotech techniques. In today's video, we will go through the topic of gel electrophoresis, the most commonly used technique in the research labs. This technique is used to separate DNA, deoxyribonucleic acid, RNA, ribonucleic acid, and proteins. The separation is carried out based on the size or molecular weight and on the basis of charge of a particular molecule in the presence of electric current. So let us learn its basic principle. The charged molecule moves when an electric current is passed across it. The negatively charged molecules, like DNA, will migrate towards the positively charged anode, while the positively charged molecules will move towards the negatively charged cathode. There are two types of gel electrophoresis: horizontal gel electrophoresis and vertical gel electrophoresis. First, let us see the basis of the horizontal one. Also known as agarose gel electrophoresis, it is mainly used for the analysis of nucleic acids, DNA or RNA. The separation occurs on the basis of size. The smaller molecules travel faster than the large ones under the influence of electric current. This is how the result of an agarose gel electrophoresis looks like. The next type is vertical gel electrophoresis, also known as SDS page. It is mainly used for protein analysis on the basis of their molecular weight. The main ingredients used are acrylamide and bisacrylamide. The proteins move according to their molecular weight in the presence of electric current. This is how an SDS page gel looks like. Stay tuned for our next video on hands-on agarose gel electrophoresis, and don't forget to like and subscribe to our channel for more such exciting videos on biotechnology techniques.